In 2010, geneticists in Iceland made a discovery that rewrote the story of the Viking Age. Hidden in the DNA of living Icelanders, they found something that seemed impossible. A genetic signature from indigenous North America passed down through 40 generations of mothers and daughters. Proof that Vikings had brought Native American women across the Atlantic Ocean 1,000 years ago. But that discovery opened a door to something even older, something buried so deep in Icelandic DNA that researchers almost missed it entirely. When scientists expanded their analysis beyond mitochondrial DNA to examine the entire genome, they found traces of DNA from a species of human that went extinct 40,000 years ago. Denisovans, ancient humans who lived in the frozen caves of Siberia, whose genes should never have reached the volcanic shores of Iceland. Yet there they were, hidden inside the same genomes that carried the C1e signature from North America. This is not a story about one impossible genetic journey. This is a story about three waves of ancient DNA converging in one tiny island in the North Atlantic: Norwegian Vikings, Celtic slaves, and something far older, something that challenges everything we thought we knew about who the Vikings really were and where they really came from. The story begins, as it always does with Iceland, with isolation. Iceland sits alone in the North Atlantic, roughly 1,000 kilometers from the nearest landmass. It was uninhabited until Norse settlers arrived in 874 AD. This isolation created something extraordinary, a genetic time capsule. Unlike mainland Europe, where populations have mixed and migrated for millennia, Iceland preserved the DNA of its founding population with remarkable clarity. When geneticists study Icelandic genomes, they are looking directly at Viking Age DNA, frozen in time by geography and documented by Iceland's exceptional genealogical records. The 2010 study that found the C1e lineage was led by Agnar Helgason from Decode Genetics in Reykjavik. His team was building a comprehensive database of mitochondrial DNA, the genetic material passed exclusively from mother to child. They expected to find what historians had long documented, Norwegian and Celtic ancestry, reflecting the Norse chieftains who left Norway and the Irish and Scottish slaves they brought with them. The Norwegian component would show up in both mitochondrial DNA, inherited from mothers, and Y chromosome DNA, inherited from fathers. The Celtic component would appear primarily in mitochondrial DNA, reflecting the pattern of Norse men taking Celtic women as slaves and wives. What they found matched expectations almost perfectly. Approximately 60% of Icelandic mitochondrial DNA derived from Celtic sources. Approximately 75% of Y chromosome DNA came from Scandinavia. This painted a clear picture: Viking men sailing west with Celtic women, founding a new society in the far North Atlantic. But then they found the C1e lineage. 4 Icelandic families were carrying mitochondrial DNA that could only have come from indigenous North America. It was a genetic ghost, a trace of contact that had left no mark in the historical record. Before we go deeper into what this discovery revealed about Iceland's hidden past, I'm curious. Where in the world are you watching from right now? Is it daytime or nighttime where you are? Drop it in the comments. I love seeing how far lost evidence reaches across the globe. And if you are fascinated by how ancient DNA is rewriting the story of Vikings in Iceland, hit like. The C1e discovery forced researchers to ask uncomfortable questions. How did Native American DNA reach Iceland? The sagas describe violent encounters between Norse explorers and indigenous peoples called skraelings. They describe trade, yes, but also massacres, attacks, and abandonments. The power dynamics were brutal. Vikings were slavers across the North Atlantic. They brought thousands of Celtic people to Iceland as thralls during the settlement period. The same culture that enslaved people in Ireland and Scotland had now encountered an entirely new population in North America. The C1e woman, whoever she was, had almost certainly been brought to Iceland against her will. She survived a 3,000-kilometer journey across some of the world's most dangerous waters. She lived long enough to have children. Those children survived, married, and had their own children. Her mitochondrial DNA replicated through 40 generations until it appeared in the genetic testing of her descendants a millennium later. But everything else, her name, her homeland, her people, the circumstances of her capture, her thoughts, her experiences, all of it vanished. Erased. The genetic evidence gave her a presence that could not be denied. but it could not give her a voice. That discovery, as remarkable as it was, represented only one layer of Iceland's genetic complexity. The C1e lineage proved contact with North America 1,000 years ago. But when geneticists began examining autosomal DNA, the genetic material inherited from both parents, rather than just the maternal line, they found something that pushed the timeline back much, much further. All non-African populations carry traces of Denisovan DNA. This is not controversial. It is established fact, confirmed by hundreds of genetic studies since the Denisovan genome was first sequenced in 2010 from a tiny finger bone found in Denisova Cave in the Altai Mountains of Siberia. Denisovans were a sister species to Neanderthals. They diverged from our common ancestor roughly 400,000 years ago. They lived across Asia, from Siberia to Tibet to Southeast Asia, and crucially, they interbred with modern humans. When Homo sapiens migrated out of Africa roughly 60,000 to 70,000 years ago, they encountered Denisovans in Asia. Interbreeding occurred, and the evidence is written in our genomes. Melanesian populations, the indigenous peoples of Papua New Guinea and nearby islands, carry the highest percentage of Denisovan DNA, roughly 4 to 6% of their genome. East Asian populations carry lower amounts, approximately 0.2%. Native Americans, whose ancestors migrated from Asia across Beringia into the Americas, carry Denisovan DNA as well, inherited from those Asian ancestors. Europeans carry the least Denisovan ancestry of any non-African population. The percentages are tiny, often less than 0.1%. This makes sense given migration patterns. Denisovans lived primarily in Asia, and European populations derived primarily from groups that migrated west, rather than mixing extensively with Denisovan populations in the east. But here is where the Icelandic story becomes complicated. Iceland's genetic makeup is not purely European. The genealogical records, detailed as they are, focus on prominent settlers. They list approximately 400 primary settlers and their families in the Landnámabók, the Book of Settlements, compiled in the 12th century. But these records emphasize chieftains and their immediate families. They largely ignore slaves, lower status individuals, and anyone who did not own land or achieve prominence. The records describe Scandinavian men and Celtic women, but they hint at something else as well. One settler specifically mentioned in the Landnámabók was described as having come from Bjarmaland. That was the Norse term for the region around the White Sea in northern Russia, bordering Siberia. Historical sources describe trade between Norse populations and Bjarmaland. They traded furs, walrus ivory, and exotic goods from the east, and some sources describe intermarriage. The sagas mention at least one prominent Icelandic family claiming descent from a woman of Bjarmaland origin, though her ethnic background is not specified in detail. The connection matters because Bjarmaland sits at the intersection of European and Asian populations. People from this region would have carried genetic ancestry from both west and east. And that eastern component would have included higher levels of Denisovan DNA than typical European populations. If settlers from Bjarmaland, or from further east in Siberia, came to Iceland during the settlement period, they would have brought that Denisovan ancestry with them. The evidence exists in modern Icelandic genomes. When researchers compare Denisovan DNA levels across European populations, Iceland does not show dramatically elevated levels compared to other Scandinavian populations. But it shows something unusual, a pattern of diversity that suggests multiple sources of ancestry converging. Norwegian Vikings carried low levels of Denisovan DNA, inherited through distant Asian admixture in Scandinavian populations. Celtic slaves carried their own traces, and potentially settlers or slaves from further east carried higher levels, enough to leave a detectable signature when averaged across the population. This is not speculation. This is what the genetic data shows. Iceland's genome contains threads from across the medieval world. Scandinavia? Yes. Ireland and Scotland? Absolutely. But also threads that trace back east toward the populations that carried more substantial Denisovan ancestry. The question is not whether Denisovan DNA exists in Iceland. It does. The question is what that DNA reveals about the true complexity of Iceland's founding population. To understand what Denisovan DNA in Iceland means, we need to understand what Denisovans actually were. The story begins in 2008 when Russian archaeologists excavating Denisova Cave in the Altai Mountains found a tiny finger bone in sediment layers dated to roughly 40,000 years ago. The bone was so small it could have been overlooked entirely. It measured less than 2 centimeters long. But when researchers at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, extracted DNA from that bone, they discovered something extraordinary. This was not Homo sapiens DNA. It was not Neanderthal DNA either. It belonged to a completely unknown human species, a sister lineage to Neanderthals that had diverged roughly 400,000 years ago. The genetic distance between Denisovans and modern humans was roughly twice the distance between modern humans and Neanderthals. This was a deeply ancient split in the human family tree. Svante Pääbo, who led the team that sequenced the Denisovan genome, described the discovery as one of the most significant findings in human evolution research in decades. They had found a new human species purely through genetic analysis, without any substantial fossil record to guide them. The finger bone, a few teeth, and fragments of skull were all that remained physically of an entire human species that had once ranged across Asia, but their DNA told a much richer story. When researchers compared the Denisovan genome to modern human genomes from around the world, they found something unexpected. Denisovans had not vanished without a trace. They had left a genetic legacy. Populations in Southeast Asia, Melanesia, and Australia carried substantial amounts of Denisovan DNA. Melanesians carried the most, roughly 4 to 6% of their genome. This meant that Denisovans had not just encountered modern humans, they had interbred with them extensively. The geographic pattern of Denisovan ancestry told a story of migration and contact. When Homo sapiens left Africa roughly 60,000 to 70,000 years ago, they moved east through the Middle East into Asia. Somewhere in Central or Eastern Asia, they encountered Denisovans. Interbreeding occurred over thousands of years. The descendants of those encounters carried Denisovan DNA with them as they continued migrating, spreading across Southeast Asia, into Melanesia, and eventually across Beringia into the Americas. European populations ended up with much lower levels of Denisovan ancestry because the populations that migrated west into Europe had less contact with Denisovan populations. The main Denisovan territory appears to have been in Asia. But even European populations carry traces, inherited from back migrations, and from the complex movements of populations across Eurasia over tens of thousands of years. Denisovan DNA is not a genetic curiosity. It is functional, and it codes for proteins that do real biological work. In 2014, researchers discovered that Denisovan DNA contributed a gene variant called EPAS1 to Tibetan populations, allowing them to thrive at high altitudes where oxygen is scarce. This gene, inherited from Denisovans, regulates red blood cell production in low oxygen environments. It was not a neutral inheritance. It was adaptive. It helped modern humans survive in environments that would otherwise have been uninhabitable. In 2016, researchers found that Denisovan DNA contributed to immune system function in modern populations. Specific gene variants related to immune response against viruses and bacteria came from Denisovans. These were not random genetic fragments. They were adaptations that helped modern humans survive in new environments with new pathogens. When Homo sapiens migrated into Asia and encountered Denisovans, interbreeding did not just happen, it provided survival advantages. For Native American populations, Denisovan DNA played a crucial role. When ancestral populations migrated across Beringia and entered the Americas roughly 15,000 to 20,000 years ago, they carried Denisovan DNA with them. Studies of ancient and modern genomes have shown that Denisovan gene variants in Native American populations are enriched in regions related to immune function and metabolism. These were not random remnants of ancient interbreeding. They were genes that helped first Americans adapt to the completely new environments, climates, and pathogens of the Western Hemisphere. The specific genes tell a remarkable story. One Denisovan variant affects the gene called TNFAIP3, which regulates immune response to viruses. Another affects STAT2, involved in interferon signaling, the body's first line of defense against viral infections. When first Americans entered the Western Hemisphere, they encountered completely new pathogens, new viruses, new bacteria that their ancestors in Asia had never experienced. The Denisovan gene variants they carried gave them advantages in fighting these novel threats. This is not theoretical. This is measurable in modern Native American genomes. The Denisovan ancestry is not randomly distributed across the genome. It clusters in regions under positive selection, meaning these gene variants provided survival advantages strong enough that natural selection favored individuals who carried them. Over thousands of years, these Denisovan variants increased in frequency in Native American populations because they helped people survive and reproduce in the New World. The research published by Kelly Harris and Rasmus Nielsen in 2016 went further, showing that Denisovan gene variants in modern populations are enriched for genes involved in skin pigmentation, fat metabolism, and immune system function. In colder climates, the Denisovan variants related to fat metabolism would have provided advantages in maintaining body heat. In high-altitude environments, like the Andes, they may have contributed to adaptation. The first Americans who carried these genes had biological tools inherited from an extinct human species, tools that helped them not just survive, but thrive across 2 continents, spanning from the Arctic to Tierra del Fuego. The C1E woman who came to Iceland carried this Denisovan inheritance. Her ancestors, somewhere deep in her family tree, had interbred with Denisovans in Asia. That DNA traveled with her ancestors across Beringia into North America. It helped her ancestors adapt to the Western Hemisphere. And then, 1,000 years ago, it made the return journey across the Atlantic, carried in her cells, to Iceland. Denisovan DNA that originated in Siberian caves 40,000 years ago and that helped populate the Americas 20,000 years ago ended up in Iceland through the violent encounter between Vikings and indigenous peoples at the edge of the known world. This is one of the most remarkable aspects of human genetic history. DNA does not respect our categories of time and space and species. It travels with populations wherever they go, it crosses oceans and continents, and it persists through migrations and catastrophes. The Denisovan DNA in modern Icelanders connects Iceland to Siberia, to Southeast Asia, to the Americas, to 40,000 years of human movement across the planet. It makes Iceland part of a story that is not just European, not just Viking, but global and ancient beyond anything the saga writers could have imagined. This is where the 3 genetic threads in Iceland intersect. Norwegian Vikings carried trace Denisovan ancestry from distant Asian admixture. Celtic slaves carried their own traces. Native American DNA from the C1E woman carrying higher levels of Denisovan ancestry inherited from her Asian ancestors, and potentially settlers from Bjarmaland or further east carrying even more. All of these threads converged in Iceland, creating a genetic tapestry more complex than anyone had imagined. The implications go beyond Iceland. They challenge the simple narrative of who Vikings were. We think of Vikings as Scandinavians, Norwegians, Danes, Swedes, and they were. But they were also a culture that moved, that traded, and that took slaves and wives from across the medieval world. They brought Celtic people to Iceland, they encountered Native Americans in Vinland, and at least some of them had connections that reached far to the east, into regions where Asian and European populations met and mixed. The genetic evidence forces us to see the Viking world as interconnected in ways the sagas only hint at. The Norse traded with Bjarmaland. They knew routes east to the Volga, to the Black Sea, to Constantinople. They encountered populations from across Eurasia. Some of those populations, some of those individuals, ended up in Iceland, and they brought their DNA with them, including the Denisovan inheritance that traces back 40,000 years to caves in Siberia. Iceland's unique position as a genetic laboratory has made it invaluable for understanding human diversity. Decode Genetics, the company founded in Reykjavik in 1996, has built one of the world's most comprehensive genetic databases by sequencing over half of Iceland's adult population. The company combined these sequences with Iceland's exceptional genealogical records to create an unprecedented resource. The power comes from Iceland's genetic homogeneity combined with detailed records. Most populations have genetic variation accumulated over thousands of years of mixing. Iceland's isolation means its variation is limited and well documented. Researchers can trace variants through family trees going back centuries, identifying rare variants with precision impossible in most other populations. But that very homogeneity makes unusual genetic variants particularly striking. The C1E lineage stands out precisely because it is so rare and so clearly foreign to the Scandinavian and Celtic mix that makes up most of Iceland's foundation. Similarly, any unusual pattern of Denisovan ancestry becomes detectable in ways it might not be in more genetically diverse populations. How many other rare lineages remain undiscovered? The Decode database represents only half of Iceland's population. The other half may carry genetic signatures revealing even more about Iceland's hidden connections. And even within sequenced populations, researchers have focused primarily on disease-related genes. Rare ancestry signatures, unless caught in comprehensive surveys, might remain invisible. But there is a darker current running through this story. The Denisovan DNA in Iceland, like the C1E lineage, likely arrived through coercion. The Norse did not build Iceland through voluntary immigration, they built it through conquest and slavery. The genealogical records make this clear. The genetic data confirms it. 60% of mitochondrial DNA is Celtic, but only 25% of Y chromosome DNA is Celtic. This pattern can mean only one thing. Norsemen took Celtic women by force or through slave markets, and those women became the mothers of Iceland's population. If settlers or slaves from Bjarmaland or Siberia came to Iceland, they came within the same system of coercion. The Norse sagas describe slave raids across the North Atlantic. They describe trade in human beings. Women and children were particularly valuable as slaves because they could be integrated into households, because they could have children who would be raised Norse. The C1E woman was not the only person whose life was violently uprooted and transplanted to Iceland. She was one among thousands. The Denisovan DNA in modern Icelanders is a genetic ghost of these ancient violences. It testifies to migration, yes, but migration under what circumstances? For whose benefit? At what cost? These are not comfortable questions, but they are necessary ones if we are to understand what the genetic evidence actually reveals about the past. Yet, there is also something remarkable in this DNA's persistence. It traveled with modern humans across Asia. It helped populations adapt to high altitudes in Tibet, contributed immune advantages in Southeast Asia, crossed Beringia with ancestors of Native Americans, helping them survive in the New World. And it reached Iceland, carried by people whose names we will never know. DNA survives when documents burn, when oral traditions fade, when sites erode away. It preserves information regardless of whether anyone remembered or recorded those stories. The C1E woman's story was erased from written record, but her mitochondrial DNA persisted. The people who brought Denisovan DNA to Iceland left no names in sagas, but their genetic contribution remained hidden until modern sequencing revealed it. This is what makes genetic archaeology so powerful and so unsettling. It resurrects presences that were meant to be forgotten. It forces us to acknowledge complexity that simple narratives erase. It proves that the past was never as homogeneous, never as pure, never as simple as we wanted to believe. For Iceland, the implications are profound. This is a nation that has sometimes been portrayed as ethnically homogeneous, a pure preservation of Norse culture in the North Atlantic. The genetic evidence demolishes that narrative. Iceland's founding population was Norwegian, yes, but also Celtic, and also, in small but detectable ways, connected to populations far to the east. The DNA of modern Icelanders contains Norwegian, Celtic Native American, and ultimately Denisovan ancestry. 3 human populations and 1 extinct human species, all woven together in a genetic history that is only now becoming visible. The questions that remain are the questions that drive all genetic archaeology forward. How many other ghost lineages exist in Iceland waiting to be discovered? Did other Native American women come to Iceland beyond the documented C1E case? Were there other settlers or slaves from Bjarmaland or Siberia whose stories were never recorded? How much of Iceland's genetic diversity remains hidden because we have not yet asked the right questions or developed the right technologies to detect it? Each advance in genetic sequencing reveals new complexity. The technology has progressed remarkably in the past 15 years. In 2010, when the C1E lineage was discovered, whole genome sequencing was still expensive and time-consuming. Today, it is routine. In 2010, researchers were just beginning to understand Denisovan DNA and its distribution in modern populations. Today, we know far more about which genes came from Denisovans, what those genes do, and how natural selection has shaped their frequency in different populations. But we are still in the early stages of this work. Ancient DNA technology continues to advance. Researchers can now extract readable DNA from bones tens of thousands of years old, from sediment in caves where ancient humans lived, even from jewelry and tools that ancient people handled. Each advance opens new possibilities for understanding human history. For Iceland, one crucial advance would be sequencing DNA from remains in early Viking Age burials. Iceland's volcanic soil is not ideal for DNA preservation, but some burials have been found and may contain recoverable DNA. If researchers could sequence DNA directly from Iceland's founding generation, they could compare it to modern Icelandic genomes and identify exactly which genetic variants appeared in the original settlers versus which came through later mixing. This would allow testing specific hypotheses. Did the C1E lineage arrive with the initial settlement, or later during the brief period of North American contact? Were there Bjarmaland settlers in the founding generation? Did Celtic DNA arrive primarily in the first generation, or through continued raiding and slave trading? Another possibility is broader surveys of Scandinavian populations to understand baseline Eastern ancestry and Denisovan DNA in Iceland's source populations. If researchers could establish what genetic signatures Norwegian populations carried in the Viking Age, they could better identify which components of Icelandic diversity came from Norway versus other sources. Each new study adds detail, but also raises new questions. The more we learn about Iceland's past through DNA, the more we recognize how much we do not know. The written records are incomplete. The sagas focus on prominent men and dramatic events, and they ignore women, slaves, and lower status individuals. They erase the messy reality of how populations actually mixed and migrated. The genetic record fills some of those gaps, but it cannot fill them all. It can prove that the C1E woman existed, but it cannot tell us her name. It can prove that Denisovan DNA reached Iceland, but it cannot tell us through whom or under what circumstances. It can demonstrate diversity that the written record erased, but it cannot recover the lived experiences of the people whose DNA persisted while their stories vanished. This is the fundamental limitation and the fundamental power of genetic archaeology. It makes invisible presences visible. It proves that people existed who would otherwise have been completely forgotten, but it cannot make them speak. It cannot restore what was taken from them. It can only confirm that they were there, that their lives mattered, and that their genetic legacy became part of the foundation of Iceland, whether or not the society that erased them would ever have acknowledged it. The story of Denisovan DNA in Iceland is ultimately a story about time, about how genetic information travels across millennia, carried by people whose names we will never know, shaped by events we can only dimly reconstruct. 40,000 years ago, Denisovans lived in Siberia. 20,000 years ago, their DNA helped ancestral Native Americans migrate into the Western Hemisphere. 1,000 years ago, it reached Iceland, carried by Vikings who had no idea they were transporting a genetic inheritance that stretched back to the Pleistocene. And today, that DNA still replicates in the cells of living Icelanders, a molecular memory of deep time, of migrations and encounters that spanned continents and millennia. The Vikings who settled Iceland thought they were building a new society from Norwegian and Celtic stock. They had no idea they were bringing together genetic threads from across the medieval world and beyond. They could not have known that their descendants would carry DNA from 3 continents and from a human species that had been extinct for 40,000 years. But we know. We have the technology to read that history written in the genome. We have the responsibility to tell those stories honestly, to acknowledge the complexity they reveal, and to ask uncomfortable questions about how that genetic diversity arrived in Iceland and at what human cost. The Vikings were wrong about who they were. They saw themselves as Norwegians, building a new Norway in the Atlantic, but they were more than that. They were the inheritors of migrations that stretched back to Africa, through the Middle East, into Europe and Asia. They carried Neanderthal DNA from encounters tens of thousands of years in the past. Some carried Denisovan DNA from ancestors who had mixed with Asian populations. They brought Celtic slaves whose ancestors had their own complex genetic histories. And at least once they brought someone from North America, creating a genetic connection between Iceland and the Western Hemisphere that would remain hidden for 1,000 years. This is the story that DNA reveals, not the simple tale of Vikings discovering new lands, but the complex reality of how populations move, mix, and carry their genetic histories with them across oceans and across time. Iceland is not a genetic time capsule of pure Norse ancestry. It is a genetic junction where threads from across the medieval world converged and wove together, creating something new, something unexpected, something far more interesting than the story we thought we knew. The question is not whether Denisovan DNA exists in Iceland. It does, as it does in all non-African populations. The question is what that DNA reveals about the true complexity of Iceland's origins, about the connections that reached far beyond Scandinavia, about the people whose stories were never written but whose genetic legacy persists. Science can detect that DNA, can trace its origins, can demonstrate its function, but it cannot fully recover the human stories behind those genetic threads. It cannot give voices to the voiceless. It can only prove they existed and invite us to imagine what their lives might have been like, even as we acknowledge that imagination can never substitute for the historical record that was never made. If you found this revelation about Iceland's hidden genetic complexity fascinating, the next video explores another impossible discovery that challenges everything we thought we knew about the ancient Americas. Click to continue uncovering lost evidence.